Hi everyone, I'm Andrew. I'm a PhD student here at Northwestern, and I'll be talking about a project I have that's going on here about spatial boundary formation. It's a collaboration between my two advisors. So there's Carol Laban, whose lab I do all of my experiments in, and Madhav Mani, who advises me on the computational and theoretical aspects of my project. So in the Laban lab, the model organism of choice is the frog Xenopus laevis. So these frogs, they can lay hundreds of eggs at a time, and they develop externally. So it's really easy to just watch development happen, which is what you see going on here in this movie. Now, the process that you see here, it's known as neurulation, where the neural plate, so the presumptive central nervous system, it will fold in on itself to form the neural tube. Now, if we opened up a textbook on Xenopus development, what we'd see is that like this process of neurulation, but also just development in general, it gets divided up just by the people who study it into several different, a handful of different stages, like temporal stages. And also the cells get divided up into a handful of different fates. So basically what a textbook would tell you is that at stage 13, which is the beginning of neurulation, the ectoderm, so the outer layer of cells that you're looking at here, it's divided into the neural plate, which again is the presumptive CNS, the epidermis on the outside, which is the future skin, and then the neural plate border in between those. And then again, what the textbook would say is that as this process goes on, you'd have within the neural plate border, you have specification of two different cell types, so the neural crest and the placodes, which together will go on to form the entire peripheral nervous system, the craniofacial skeleton, pigment cells, kind of a whole wide variety of different cell types. And one interesting thing to note about those is that even though they kind of form structures in the adult organism that are not necessarily vertebrate specific, these two cell types are unique to vertebrates. So it's a unique aspect of vertebrate development that we're studying. So right away, we can just kind of, by comparing these two different views, the movie on the left with the pictures on the right, we can see that there's kind of like a disconnect between them. So when we actually watch development happen, like in this movie, what we see is kind of a relatively smooth and continuous process in time. And then when we compare it to kind of what's, the fixed stages and the fate map on the right, something that like a simple fate map, like what we see on the right, lacks is kind of spatial and temporal resolution. So there's really no indication about what might be going on with just individual cells that are kind of found at the interface of these different fates. So when I joined the lab, I started using fluorescent whole embryo imaging techniques to start addressing that question of what's actually going on at the interface of the neural plate and the neural plate border. So in this image here, it's from a mid-neurula embryo, so stage 15 approximately. I just performed an immunostain, so I used an antibody to detect the protein of these two different transcription factors that are commonly used as fate markers. So SOX2 is what you see in green here that marks the neural plate, and PAX3, which is in magenta, that marks the neural plate border. So PAX3, it marks the neural plate border at early stages, and then as neural crest is being specified throughout neurulation, it's thought to be restricted to the neural crest. So it's kind of a border and neural crest marker. Basically, we would have thought that by stage 15, that these two fate markers would be separate if the neural plate and the neural plate border have distinguished themselves by this point. But it turns out when we actually zoom in on what's going on at this interface of these two different fates, we still see a significant number of cells that co-localize these two transcription factors. So there are a few that are indicated by arrows in that image, but then there are also several more that's kind of interspersed throughout. So we do see lots of co-localization of these transcription factors. And kind of another way of stating this observation is that just at this stage, these transcription factor boundaries overlap in space. But if we just look a few hours later in development, so if we look at stage 17, which is at the end of neurulation, or like near the end of neurulation, these fate markers have completely segregated. We no longer see co-localization of these factors, and we even see this narrow band of cells in between where we don't detect either of these proteins. So kind of another way of just stating this observation is that by this stage in development, just at the interface of the neural plate and the neural plate border, this boundary has resolved. So boundary resolution processes kind of like this, they're pretty common throughout development, and they're frequently studied. And kind of one of the paradigms of the field that's popped up is that a lot of times mutual transcriptional repression is what's responsible for resolving the boundary between transcription factor patterns like this. So even though in these images I'm only looking at one particular marker, one transcription factor for each of these fates, behind each of these fates there's a complex genetic regulatory network going on. And we hypothesized that kind of something within the neural crest network might be it's probably responsible for repressing something within the neural plate network and vice versa. So that's kind of the hypothesis that I started out this project with. So I quickly just want to show you kind of what a more traditional Xenopus project would look like to start addressing that question. So one of the real benefits of Xenopus as a model system is that the first cell division actually splits the left and the right halves of the embryo. So this is incredibly useful for experimentalists because we can just inject one cell with a construct that we want to overexpress, so we can manipulate development on one half of the embryo only, while the other half kind of develops normally and just serves as a perfect internal control. So in this experiment here, I just started with the candidate gene slug. So it's an early specifier of neural crest tissue, and it's also been shown kind of in other contexts of development to directly repress SOX-B1 factors, so SOX2 and SOX3 is repressed by slug, and again, those SOX2 and SOX3 are specifiers of the neural plate. So it seemed like a good candidate gene to start with. So what I did in these experiments is just injected RNA that codes for the Xenopus slug gene, so X slug, which you see in this top row here, and then a dominant negative construct known as delta slug in this bottom row here, which is thought to knock down the functioning of slug. And in all these images, the injected side, so it's the red area kind of marked by beta gal, so this serves as a lineage tracer just so we know which side of the embryo was injected and manipulated. So basically, if this model that we have over here of boundary resolution, if that's correct, then we'd expect that overexpressing a neural crest specifier would reduce the neural plate, while knocking down the neural crest specifier would expand the neural plate. And when we look at the effects of the neural plate by looking at SOX2 RNA, which is shown in this chromogenic in situ here, that is basically what we see. So inject slug, overexpress neural crest, then we see kind of a loss. Basically, this side is in this side of the SOX2 embryo is not as dark. And then when we knock down neural crest, we see kind of a spatial expansion of that. But I think kind of what's really interesting here is when we look at the distribution of PAX3, because in both cases where we're doing these two opposite manipulations, overexpressing slug or knocking down slug, we observe the same phenotype. So there's a spatial expansion of kind of the PAX3 region on the injected side relative to the uninjected side. So with this, already we're kind of coming up against the limitations of this traditional chromogenic in situ technique. So it's difficult to look at more than one gene in a single embryo. You can't really do it. You can do two-color in situs, but it's kind of difficult to get meaningful data out of that sometimes. So just the fact that you can really only look at one gene at a time makes it difficult to use for addressing questions of this overlap between patterns and resolution of boundaries like that. Another thing is that this technique is not particularly quantitative. So for example, in PAX3, it's possible that, in this region, maybe it's spatially expanded, but it's also possible that the abundance of transcripts has decreased slightly. It's not really a judgment we can make from that. And kind of finally, these are limited to just detecting RNA. So if transcriptional repression is at play, we should be able to kind of see the effects on the level of RNA. But at the end of the day, for these transcription factors that we're studying, what matters is just how much protein is present in a given cell. So another reason why we think that just looking at RNA patterns might not be enough is that for some of the genes we're interested in, their patterns at the RNA and the protein level are actually qualitatively different. So these images here are from two different embryos. So the RNA image on the left, this shows the RNA distribution of SOX2 in an embryo. I obtained that using a technique for fluorescent in situs known as hybridization chain reaction, or HCR. And then on the right, the image of the protein obtained using immunofluorescence. So it's an antibody that recognizes SOX2. So this image on the right, the protein image, if we kind of look at the periphery of the SOX2 region, what we see is more or less like a smooth drop-off in intensity at the periphery. But then when we look at the RNA image and compare that, something I kind of see again and again in these RNA images are these more lateral stripes of the lateral stripes of the SOX2 region. So we see this kind of very bright SOX2 region in the middle, followed by a band of maybe one or two cells that are dark, then followed by another band of 2 or 3 cells where we detect more SOX2 RNA. And that's just, by looking at these two and comparing them, that's kind of very qualitatively different from what we see in the protein case. And even though I'm just showing you SOX2 here, we have noticed similar kind of discrepancies in RNA and protein looking at PAX3 also. So what this kind of suggests is that the levels of these different transcription factors, they might not just be regulated through transcription. So that model we had earlier of just transcriptional repression resolving this boundary. It might not be that simple. And it also kind of highlights this, just like highlights the need to image the entire central dogma to start addressing this boundary formation question. So just on the topic of imaging the entire central dogma, there's kind of one more step of transcription we need to image. So one thing that we want to be able to do is just differentiate between cells that are actively transcribing RNA versus cells where, we're still detecting RNA with these FISH probes, but maybe just because the transcript is long-lived and, it might have turned off, the cell might have turned off transcription, but the transcripts are just still there. That's kind of something we want to be able to differentiate between. So for PAX3 specifically, we had these FISH probes that were designed against the intronic regions of the sequence, which you see here in the green channel. And I want you to note that these probes only detect these bright sites of, these bright puncta, so these sites of transcription that are associated with nuclei. So we look at the merge with DAPI and we see that these kind of bright puncta, which likely are sites of transcription, are each associated with the nucleus. And then but the thing is, like when we look at the exonic channel, so the image with probes that are only designed against exonic regions, we see that these, bright puncta are redundantly detected with probes that are just designed against the exonic regions of the gene. So kind of using these separate exonic and intronic probes for PAX3, I wanted to collect data that might start giving me an idea of how individual cells might be tuning their levels of PAX3 just through transcription. So this transcription is the starting point for this, but I want you, I think you'll see that what I'm about to show, it's not just limited to that. So you'll hopefully see that this technique can be extended to look at protein levels as well, and also to look at multiple genes in parallel, even though so far I've only done it for PAX3. So really what we're limited by is just the number of distinct probes that we can image at one time. So for this experiment I'm about to show, I collected 10 embryos per stage at 4 different stages throughout neurulation, and then just took zoomed in images on of the PAX3 positive region on both the left and the right halves of the embryo. And these, again, are just in wild type embryos here. So just following the collection of the images, to get actual data out of these images, I developed an image analysis pipeline to quantify these, the intensities that are associated with the exonic and the intronic signals. So first, we just detect the surface layer of cells that I'm interested in with these 3D confocal Z stacks. And then detect that surface and then map that to a flattened surface. So afterwards, we segment individual nuclei and then use that nuclear segmentation to make what's called a Voronoi diagram to approximate cell boundaries. So I'll remind you that the exonic RNA signal, it's diffuse throughout the cytoplasm. So a nuclear marker isn't necessarily good enough to get a single cell look at that. So we need some way of approximating cell boundaries. So that's why I use the Voronoi diagram to do that. Now, once those boundaries have been drawn, we can just label each nucleus and its corresponding cytoplasm, and then finally make mean intensity measurements within each of those regions. So you might remember from the images of the intronic probes that detect nascent RNA, so it kind of RNA is being actively transcribed. We only detected that in the nucleus, but then we also redundantly detected those puncta with the exonic probes, and that those puncta were associated with individual nuclei. So what I found, which I'm not showing here, but will likely be a powerful tool as we move forward, is that we can actually estimate the relative levels of nascent and mature RNA by comparing the nuclear just using exonic probes and comparing the nuclear signal of those exonic probes to the cytoplasmic signal of the same probes. So this plot here, it just takes intensity measurements from the exonic PAX3 probes, but it plots the nuclear signal against the cytoplasmic signal for each cell. So again, like I said, we can use that just as a proxy for nascent RNA abundance versus mature RNA abundance. And there are a few key features of this plot that I want to point out. So at all these stages, so I did 13 through 19, just 4 stages within there. This kind of cloud of high expressing PAX3 cells, it's biased towards the nuclear signal. So what this hints at is, it's something that we can follow up later on down the road with other experiments, is that these PAX3 transcripts are probably relatively short-lived, which, rapidly, short-lived, rapid transcripts with rapid turnover times, that would have implications on its own for dynamics of pattern formation. Now, the other feature I want to point out is this cluster of cells that kind of appears at later stages. So we see it in 17 and 19. So these cells that have high cytoplasmic signal but a low nuclear signal. What this suggests is that there likely are cells that are turning off transcription. So I guess And the next question to follow up on these findings that I'll be working on is connecting this back to the spatial data that we have and just asking, are the cells that turn off PAX3 transcription those same cells that we see at the interface of the neural plate and the neural plate border? And then how is that related to the resolution process that we see between these two different transcription factors? So moving forward, this is something I'll be able to use not just for one gene, but to compare multiple genes and the overlap between those and how that kind of resolves over time. So like I said, I do all of my experiments in the Laban lab, so I like to thank all of those members for their guidance and training me in those techniques. I like to thank Madhav Mani and everyone in his group for the advice I've gotten on that part of my project. And yeah, I'd like to thank my funding source and my committee as well. Thank you.